and welcome to another episode of Katie the Science Lady. I'm Mrs. Jacobson, and today's topic is part 2 of protein synthesis, translation. So let's learn together. All right, guys. So we are going to start talking about the second part of protein synthesis, translation. In our previous video, we talked about how we need a copy of DNA. So we make mRNA from DNA as just a single-stranded copy so that our genetic information can leave the nucleus. DNA is normally trapped there. So we're going to start this by knowing we've made mRNA, it's left the nucleus, and it's on its way to a ribosome. So here's our quick overview. In translation, that ribosome uses the mRNA to put together a sequence of amino acids. If we remember back to when we talked about biomolecules, amino acids are the building blocks of proteins. So this should all make sense. It should all be coming together now. We are going to use these amino acids to build a nice long chain called a polypeptide, which will become a protein. So a couple of key things to remember here. These may be new for some of you. mRNA gets read 3 bases at a time, so 3 letters at a time. Remember, we're using the letters A, U, C, and G. We're in mRNA now. There's no more thymine, so A, U, C, and G. Each of the 3 bases is called a codon. So each set of 3 bases is 1 codon. 1 codon is going to equal 1 amino acid. So you can also say that 3 bases will be 1 amino acid. That's also fine. You're going to hear me say the word codon a lot, so just keep remembering 3 bases is a codon. So from this mRNA, we can separate it into codons by grouping 3 bases at a time. So UAU is our first codon, and that's going to represent amino acid 1. CUA is our second codon, and it'll represent amino acid number 2. And GGU is our third codon here, and it will represent amino acid number 3. Now, they're going to have specific names, and I'm going to take you through that in just a moment. In order to figure this out, we have to use a codon wheel or a codon chart. I'll show you using the wheel first, and then after that, I'll show you how to use the chart. So we take UAU, our first codon, and what we're going to do is start in the middle and work our way outward. You want to move the shortest distance, but not crossing any one of these kind of big lines here, the quadrant lines. So I'm always going to go outwards to the closest one. So I start here, U, find the closest A, which is going to be right here, A. You only want to go across one line, basically. So if I go here, I'd have to cross 1, 2. If I went over here, I'd have to cross 1, 2. That's too far. So U, A, and then the next U, which is here. And that's going to give me tyrosine. So if I take UAU, it will give me tyrosine. For our second one, CUA, let's do that in our codon wheel. We take C here, go to the closest U. Now I have to go outward. I can't go up this way. I have to go outward in our spokes of our wheel. So C, then I go to U, then I go to the next closest A here outward, which is leucine. And that's our second amino acid. Our third is GGU. So that's our third codon that we need to look at. So I'm going to start here with the G. Go to the closest G outward here, and then I'm going to go outward to the closest U, which is going to give me glycine. So that's how you use a codon wheel. Again, that was just it a little bit bigger. Codon charts are a little bit different. I think for some of us they're easier, for some of us they're more difficult. I learned both ways when I was growing up, but it's kind of whatever floats your boat, honestly. If I'm going to find, let's say, AUG, I need to first look at my first letter. So first letter is A. My second letter here is U, so I go to where they meet, which is this box here. And then I find AUG in that box, which is methionine. You can also just search for your codon in this giant chart if you wanted to, but it's kind of a waste of your time. It's easier to learn how to actually view the chart. So let's say I wanted to find UGG. My first letter is U. My second letter is G over here. So I need to be in this box because it's where it intersects. And then G down here would be tryptophan or TRP. They're mostly written in their first 3 letters or 3 of the letters, and everybody knows them like that. But UGG is tryptophan. we have a couple of special codons, some that we may have to memorize or some that are just really important to this process. At the first thing, we have a stop and a start codon. And the start codon is one that you should probably memorize. There's only one start codon, which makes it unique. There are 3 or 4 stop codons, so those you may not have to memorize, but it might be a good idea. Our start codon is AUG, and this is something that I remember pretty quickly. It may differ if you're in a different country or in a different state, but where we're located, school starts in August. So that's how I remember this. It's a start codon. We start school in AUG for August, so that's a good way for me to remember it. If you start school at another time, you're just going to have to memorize it. I don't have a good way for you to memorize it other than that. So if we're looking at our mRNA here, remember that's our start codon. So anything before the start codon is not part of our protein. It's just part of the DNA and then the mRNA. So here we are with our mRNA. Our amino acids, we have to go through and we don't start, methionine is our start codon, until we hit AUG. These are 2 other codons here at the beginning, but they don't count towards our protein. Our cell only knows to stop, our ribosomes know to stop when they hit a stop codon, and you'll see them marked as stop on your chart. They don't have actual names, they're just stop codons. And in this case, UAA is our stop codon. So AUG is a start. We have our actual protein here, which would be these in-between codons, and then we hit our stop codon, which is where we're going to stop. Another way to see this, you have AUG as your start codon. It's always your start codon. You'll have as many of these internal codons as you need to make your protein. And then at the end of that gene in the DNA, you've got what codes for a stop codon. So that's what you're going to stop with. Here's this quick process one more time. I'm going to run through it again. This is the entire process of protein synthesis. And I think this is a really good way to visualize it. It's just helpful. So we start in the nucleus. Transcription happens in the nucleus. And all transcription is making a messenger, a copy of DNA. We call that messenger mRNA, and it means messenger RNA. Now that we've made this nice skinny single-stranded piece of genetic information, it can leave the nucleus. It fits through those pores, and it can leave and go to the cytoplasm where it attaches to a ribosome. When it's at the ribosome, translation occurs. It moves along till it finds the start codon, then it starts translating each of those codons until it hits the stop codon. In the next slides, I'm going to talk to you about how translation actually happens. We know we start with the start codon, AUG, and we end with the stop codon, but I'm going to fill in that in-between part. And we make this polypeptide chain here, which will be folded into a protein. So here's how it works. We have to figure out how our ribosome gets what it needs. How does it get those amino acids? The answer is that a tRNA molecule is going to bring amino acids to the ribosome. So let me kind of break it down. Every codon of our mRNA matches with the anticodon, or the bottom part, of a tRNA molecule. So the tRNA is called transfer RNA, and I think of it literally like a transporter. Its job is just to bring amino acids to the ribosome and then leave. And it knows that it's connected to the right one if the mRNA codon, the 3 bases, matches the anticodon, the bottom part of that tRNA. So a codon's like one single ingredient. tRNA is bringing the correct amino acids in the right order. So it's just a delivery truck, basically, transfer RNA or transport RNA. For every single codon that exists, any combination of 3 letters, there is a single tRNA that matches it. So the tRNA and the amino acid, they're connected. And every time you have a different codon that's going to ask to bring an amino acid, let's say I have AUG, and it's a start codon. There's an amino acid called methionine that is coding for the start codon. So if I have AUG, my tRNA is going to come on in and match with it, and attached to that tRNA is going to be my amino acid methionine for start codon. So here's kind of a visualization of that. I think this may help a little bit more. You have your mRNA codon down here. It's GAG. This molecule up here, this purple end of the molecule, it's actually much, much bigger, but it's a tRNA molecule, and this is the bottom of that molecule. That's, you know what I mean, bottom of the molecule. And at the bottom, there is an anticodon. It's just pairs that match up with that codon in mRNA. They match up so that we know we're bringing in the correct amino acids at the correct time. So attached to this tRNA is an amino acid. So here's essentially what happens. As every single amino acid gets brought to us by the tRNA, the transfer RNA brings us amino acids, we connect them with a peptide bond. So the ribosome helps form a peptide bond, a very strong bond, between each amino acid, linking them in a chain. So we see that here. We've got, these are the tRNAs, these kind of like soup can looking things. That's tRNA. And then everything off of it, these little purple molecules here, those are amino acids, and they are linked with strong peptide bonds. And that's what we're looking at here. You can also see the codon for the part of the mRNA here, and the anticodon here at the bottom of the tRNA molecule. Now, floating in your cytoplasm, you have tRNA, which is attached to amino acids. They're just ready and waiting. So as soon as you have a section here of the mRNA, anticodons will start to match up with it and bring in those amino acids. This is another visual over here of an amino acid sequence, also called a polypeptide. It's called a polypeptide because poly means many, and peptide is the type of bond between our amino acids. So many peptide bonds is basically what it's called. Now that polypeptide is not a protein yet. So what has to happen in order for it to turn into a protein, it has to be folded in a couple of complicated, complex ways. And what you're seeing here is actually a protein called hemoglobin that is going to deliver the oxygen to all of the cells in your body. So it's part of your red blood cells. And it's a protein called hemoglobin. Again, I want to just reiterate the process one more time. Let's take a deep breath. So down here we have our mRNA that has left the nucleus and found a ribosome, which is this gray structure in the picture. Every time we have 3 base pairs, that's called a codon. A tRNA molecule, which is this kind of beakery looking thing, is going to match up with each codon based on that sequence. So it has an anticodon on the bottom that matches. As the tRNA comes in, it brings an amino acid with it. It's already attached. Now, every time you bring in a new amino acid, it gets transferred and added to this growing chain called a polypeptide. It's a pretty complicated process, but as you look at more diagrams and you hear about it more, it becomes a little more clear. One quick review here, the types of RNA. We've talked about 2 types of RNA so far. We talked about mRNA, which is our messenger RNA. It's single-stranded. Its job is to just take our DNA copy to the ribosome. We have our tRNA, our transfer RNA. Some people call it transport RNA, and I understand why, but technically it is transfer RNA. It has the anticodon on one end and the amino acid on the other, and its job is just to deliver amino acids. And our third type of RNA is something you probably haven't heard of before, but it's ribosomal RNA, and it is what the ribosome is made of. So all 3 types of RNA are used in protein synthesis. mRNA is created in transcription, it's read in translation, and tRNA help build a polypeptide in translation. rRNA just makes up that ribosome where translation happens.
Let's review what we've learned so far about translation. Translation is the second part of the process of protein synthesis. And translation starts at the ribosome. So our mRNA molecule makes its way to a ribosome where it settles in between the subunits. The subunits read each codon until finding the start codon, AUG. Then, tRNA brings in 1 amino acid for each set of 3 letter bases, also called a codon. Each codon equals 1 amino acid. As the amino acids are dropped off by the tRNA, peptide bonds are formed between them. These are strong bonds and create a polypeptide chain of many amino acids together. When the stop codon is reached, this polypeptide is released, and then it folds into a protein by having different methods of folding. Once that protein is folded, it is released to the cell, and it can have one of hundreds of thousands of jobs within the cell that help us operate. That's it for today. I hope you had fun. I hope you learned something, and I'll see you later.